division. The life of all multicellular organisms starts as a single cell. According to cell theory, every cell arises from the pre-existing cell. So, growth and development of every living organism depends on cell division. So, cell division is a process by which a cell divides into two or more daughter cells. A series of changes that occur during the life of a dividing cell is known as cell cycle. The generation time is the period required for a cell to complete one full growth cycle. It is the period from the beginning of one cell division to the beginning of the next cell division. The cell cycle has two main phases. First one, interphase. It is also called a resting phase. It is highly active and prepares itself for the cell division. So now it is preparatory phase. It involves many metabolic activities. There are three phases in interphase. First one, G1 phase. It is the first growth phase. During G1 phase, following changes take place: synthesis of proteins and enzymes, production of ATP molecules needed for DNA replication. Then G0 phase or quiescent stage. The changes during G0 phase, cell may undergo G0 phase when it withdraws from the cell cycle. A cell in G0 phase either enters into G1 phase or is differentiated into a permanent cell. In the case of cancer, there is loss of control at the G0 phase and therefore it shows uncontrolled cell division. Then S phase. The changes during S phase: replication of DNA, entire genetic material or DNA duplicates during this phase, nucleus doubles in size, histone proteins needed for organization of chromosomes are formed. Now next phase, G2 phase. It is growth phase. The changes during this phase are: cell organelles such as mitochondria and chloroplast are duplicated, the centriole duplicates, tubulin, the simple protein and RNA are produced, and the cell prepares itself to enter into mitotic phase. Now the types of cell division. In prokaryotes, the cell division is atypical, that is binary fission. In eukaryotic cells, the division is of following three types. 1. Amitosis. Nuclear membrane is retained throughout. The nucleus enlarges, elongates and divides directly, followed by the division of cytoplasm. This forms two daughter cells. Amitosis leads to the structural and functional irregularities as chromatin material is not distributed equally to the daughter cells. The example, paramecium. Then mitosis. Here chromosome number remains the same. The genetic material is equally distributed to the two daughter cells. Division of nucleus and cytoplasm takes place only once. Mitosis is essential for growth, development and repair of wear and tear of multicellular organisms. It takes place in somatic cells. In animal cells, this is known as centric division, and in plant cells it is called acentric division. Mitosis involves two divisions. First one, karyokinesis. Here the nucleus divides into two daughter nuclei. It is subdivided into four phases. First one, prophase. The chromatin network condenses and appears in the form of definite number of chromosomes. Thin and long single-stranded chromosomes start condensing and get converted into diodes. The chromatids become thick and short. The nuclear membrane and nucleolus disappear. Daughter centrioles take polar positions, and prophase is the longest phase in mitosis. 2. Metaphase. Condensation of chromosome is complete and thick chromosomes are organized along the equatorial plane. Kinetic spindle is formed. Astral mitosis is seen in animal cells, while anastral mitosis is seen in plant cells. Third, anaphase. It is the shortest phase of karyokinesis. Centromeres divide into two and formation of daughter chromosomes is completed. Kinetic spindle has 4 types of fibers. 1. Astral rays. Here rays radiating away from the centriolar pairs towards the periphery of the cell. They are in animal cells only. Then interpolar fibers. The fibers extend between the opposite pairs of centrioles at the polar ends. Chromosomal fibers, they connect the centromeres to respective poles. The interchromosomal fibers appear between the centromeres of daughter chromatids. Then, telophase. Daughter chromosomes undergo uncoiling and hydration to form chromatin network. The nuclear membrane reappears around the daughter chromosomes, forming 2 daughter nuclei. Spindle fibers disappear and nucleolus reappears in newly formed nucleus. Then, cytokinesis. It is the division of cytoplasm. In animal cells, it is known cleavage. During this, the furrow formed in the plasma membrane deepens, forming 2 daughter cells. In plant cells, there is formation and extension of cell plate from center to the periphery. Now the importance of mitosis. 1. It is important in growth, development, healing and repair. 2. In the maintenance of chromosomal number. 3. It shows equal distribution of the genetic material. 4. It maintains the genetic characteristics. It is important for asexual reproduction. Now we'll turn to meiosis. It is a complex type of cell division. Meiosis or reduction division occurs only in the diploid cell. Chromosomes duplicate once, but the cell divides twice during meiosis. One parent cell forms 4 haploid cells. There are 2 successive divisions. The first one, meiosis 1. Under this, first is interphase. It is a preparatory phase in which duplication of genetic material takes place. Then, karyokinesis. It consists of prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 is the longest phase. It is subdivided into 1 leptotin. Here chromatin network forms long, thin and thread-like chromosomes. Then zygotin. Homologous chromosomes begin to pair in lengthwise manner. This pairing is known as synapsis and the pairs of chromosomes are bivalents. Then pachytene. The chromosomes become short and thick. Bivalence. Homologous chromosomes become compact and result in breaking and reuniting of chromatids known as crossing over. Here exchange of genetic material and recombination takes place. This leads to variation. The points at which crossing over takes place are called chiasmata. Then diplotin. Here homologues separate out and repel from each other, but they remain attached at chiasmata. Nucleolus and nuclear membrane start disappearing. Then dikinesis. Chromosomes continue to condense and shorten. The homologs separate and chiasmata are shifted to the ends of the chromatids. It is terminalization. The nucleolus and the nuclear membrane completely disappear and the kinetic spindle becomes clear. Now metaphase 1. Bivalents rearrange on the equatorial plane. Spindle formation is clear. Chromosomal fibers extend from the polar ends to the corresponding centromere. Anaphase 1. Chromosomal fibers contract and pull the homologs. They shift to the opposite poles. The centromeres remain undivided. Chromosomes finally get separated. Process is disjunction. By the end, half of the chromosomes are seen at the one pole and other half at the opposite pole. Then telophase 1. Chromosomes uncoil and form chromatin. The spindle fibers disappear. The nucleolus and the nuclear membrane reappear and two daughter nuclei are formed. Cytokinesis 1. The plasma membrane constricts in the middle of the cell and two daughter cells are formed. In plant cells, cell plates are formed. Each daughter cell is with haploid set of chromosomes in its nucleus. Then interkinesis. A very short resting period between meiosis 1 and meiosis 2 is called interkinesis. It may or may not be present. Now meiosis 2. In this, prophase 2, chromosomes and chromatids become distinct, nucleolus and nuclear membrane disappear. Then metaphase 2, spindle formation takes place, chromosomes move towards the equatorial plane, chromosomes get connected to the respective poles by the chromosomal fibers. Then anaphase 2, centromeres and chromatids of every chromosome divide, interchromosomal fibers are formed, chromosomes move towards the opposite pole. Telophase 2, daughter chromosomes which are moved towards the poles start uncoiling and form chromatin network. Nuclear membrane and nucleus reappear. Two separate daughter nuclei are formed which have haploid number of chromosomes. Cytokinesis 2, division of cytoplasm forms two daughter cells. At the end, four daughter cells are formed. Now, importance of meiosis. Sexual reproduction is possible by the process of meiosis. Chromosome number is reduced to half. Genetic recombination is produced. Variation required for the evolution is possible.